My name is Joe Abisambra. I do Alzheimer's disease research. So the disease was described over 100 years ago, and we still have no cure. This is a very exciting time to be in science, not only because we have really new and exciting tools, but also because the world is a lot smaller. And because of the use of technology and forums and that sort of an element that didn't exist before, we can now communicate, not only with people on the same floor, but also around the world. It used to be in old scientific buildings that you would see silos, individual silos where you have independent labs working on their own independent things. Today what we have is this open hall of bench after bench after bench of many different labs. The whole goal of the building is to bring science together to cure Alzheimer's disease. Every single individual here is 100% dedicated to finding a cure. So here we have the lab of Dr. Kong. Something to the lab of Dr. Jinwell.
Hi, how are you?
Opening into Dr. Dickey's lab.
Hi.
You see all the tools and equipment. Do you need more light? They're right in here. So obviously the brain of a mouse is much smaller, and here we've actually microdissected a small structure of the brain called the hippocampus. As what we've done is we've taken our drug, injected it here in the hippocampus. This is to prove that it's expressing. The problem in this experiment was that we were getting the drug in neurons, but not in every single neuron. So then we tested with microscopy where our drug was, here in green, where the tau pathology was, the bad protein in disease, and we found that the cells that had the drug did not have the tau. We got rid of the pathology, which is green. And then to test that, we did the electrophysiology experiments. And as you can see here, this at 0.0 represents when we shock them with electricity, or when I scream at the cells. And you can see in the black signs, whether it be a rectangle or a triangle, are sick mice treated with our controls, placebo. In the inverse triangles in white, you see a normal mouse. And how it remembers, and over an hour, it still has a pretty strong potentiation. Our sick mouse with the drug is in the circle, and you can see that it traces almost perfectly along the same path that the normal mouse is. So effectively, with this treatment, we've rescued the brain function. So one of the great advantages of being at the Byrd Alzheimer Institute is that we're primarily an academic institution, and our goal is to find cures for Alzheimer's disease. The idea now is that we're not tied down to finding something that we can patent. For example, there are reports of certain compounds like caffeine or dyes that can help in these diseases. So nobody can patent caffeine, which means that we can work with caffeine. Nobody else will probably be interested in it. And in fact, we have publications showing that increased levels of caffeine on a daily diet can improve the brain health and stave off Alzheimer's disease.